A student designed an experiment to determine if air temperature had an effect on the rate of photosynthesis in corn plants. The independent variable in this experiment is the 1, air temperature at which the corn plants were grown, 2, amount of carbon dioxide used by the corn plants, 3, volume of oxygen produced by the corn plants, or 4, the number of corn plants used. Show me what you got in 3, 2, and 1. All right, can I hear first from Anissa, please?
I got 1 because on the graph the independent variable is on the x-axis, and on the x-axis is where the air temperature is.
Okay, perfect. If you didn't have a graph, how could we use this sentence right here to give us the answer? Anissa, finish that up. So remember, we always look for that magic sentence. Okay, how do we find the independent and dependent variable using that sentence?
When it says it had the effect on the rate of the photosynthesis.
And that would be the what?
The dependent.
Dependent, right? So you're looking for air temperature had an effect on the rate of photosynthesis. So air temperature is going to be your independent, rate of photosynthesis is your dependent. So the correct answer here, independent variable is 1. Great job, Anissa. All right, number 2, a student wondered if butterflies would show any differences in their wing color if as caterpillars they were grown in the dark or grown in bright white light. Which statement would be a possible hypothesis for an experiment to test this idea? Number 1, caterpillars exposed to bright white light will show more blue and green in their wings when they become butterflies. Number 2, will caterpillars kept in the dark have brighter wings when they become butterflies than caterpillars exposed to bright white light? Number 3, 10 caterpillars will be kept in the dark and 10 will be exposed to bright white light and allowed to develop into butterflies. Or 4, results show that caterpillars kept in the dark and those exposed to bright white light had the same wing color when they became butterflies. Show me what you got in 3, 2, and 1. All right, can I hear from Ariana for this one, please?
I said it was 1 because it says the number 2 is the question, number 3 is like the experiment, and number 4 is the conclusion. So number 1 would be the only one that's reasonable.
You love that process of elimination. Thank you, Ariana. So number 1 here is your hypothesis, because as Ariana said, number 2 is your question, number 3 is a procedure, and number 4 is your conclusion. All right, number 3, a student performed an experiment to see if water temperature affects the level of activity in aquatic snails. The student set up 4 tanks with 5 snails in each tank. All 4 of the setups were identical in every way except for the temperature of the water. In order to make the conclusions more valid, the student could 1, alter the pH of the tank, alter the pH of the water, 2, change the size of the tank, 3, carry out the experiment for a shorter period of time, or 4, use a larger number of snails. Show me what you got in 3, 2, and 1. All right, can I hear from Karen for this one, please?
It's number 4 because the sample size is just 5 snails, and to make the conclusions more valid, they will have to repeat the experiment, and using a larger number of snails would get that done.
Absolutely, that was perfect. So remember, there are 2 ways to make something more valid. Karen just gave us both. You can repeat the experiment, you can do more trials, or you can use a larger sample size, so use more subjects. In this case, we're going to use a larger number of snails. Perfect. Number 4. Base your answer to the question on the information below and on your knowledge of biology. Students in a high school biology class conducted an investigation on pulse rates. The 30 students performed 3 different activities and determined their pulse rates. Each activity was done 3 times. The average is shown in the graph below. The students want to improve the validity of their conclusion. The best way to accomplish this is 1, change the hypothesis, 2, repeat the investigation several times, 3, increase the number of variables, Or 4, increase the height of participants in each group? Show me what you got in 3, 2, and 1. All right, can I hear from Christian, please?
I got 2 because the question is asking like what could you do to make the conclusion more valid, or well, yeah, and then repeat the investigation or experiment is what the answer is, since it's gonna you could check if your answer is gonna be consistent.
Absolutely, right? You're checking for consistency. You're checking to see if you got the right answers multiple, or if you got the same results multiple times. So that's going to improve your validity. Great job. All right, number 5. In an appropriately designed experiment, a scientist is able to test the effect of 1, a single variable, 2, multiple variables, 3, the hypothesis, or 4, scientific observations. This is a tricky one. Show me what you got in 3, 2, and 1. All right, can I hear from Noreen for this one, please?
Scientists are only able to test the effect of one variable on another variable.
Absolutely, and what variable are we talking about when we're discussing that one variable?
The effect of the independent on the dependent.
Absolutely, so we're looking at the effect of the independent variable. You can only test one independent variable at a time. So we're only looking at the effect of a single variable. Thank you, Noreen. Any questions about numbers 1 through 5 so far? All right, let's move on to number 6. Which activity would be an appropriate first step when designing an experiment? Number 1, reporting a conclusion based on multiple experimental trials. Number 2, researching the problem using information from a variety of sources. 3, creating a data table to organize experimental observations. Or 4, repeating the experiment with a different hypothesis. Show me what you got in 3, 2, and 1. All right, can I hear from Benjamin for this one, please?
So for the number 1, number 3, and number 4, they're all steps you usually do later on. And the first one that you usually do is research the problem, and then is the hypothesis. So number 2 would be correct.
Absolutely. So the only one that you should do first before designing an experiment is research that problem. You can't report a conclusion, create a data table of your observations, or repeat the experiment until it's already done. So the thing that you should do first is research that problem. Concentrating during class. They thought it may have some connection with the fact that these students consume energy drinks just before class. An experiment was proposed to find out if there is a connection between energy drinks and the lack of ability to concentrate in class. A properly designed experiment to determine this would include having 1, the whole class drink energy drinks and no water at all for the entire time of the experiment. 2, the whole class drink water and no energy drinks at all for the entire time of the experiment. 3, the students drink both water and energy drink just before class. Or 4, half the students drink water and the other half drink an energy drink just before class. Show me your answers in 3, 2, and 1. All right. Can I hear from Ricky for this one, please? Ricky, are you there? All right, Faraz, take over for him.
I picked 4 because in an experiment you have to have a control group and an experimental group. So half the students will water, and the one that drinks the energy drinks is going to be the control in the experimental.
Perfect. So remember, it's important to have a control group so you can compare your results to someone. So in this experiment, the students who are drinking water are the control group, and those drinking the energy drink are the experimental group. Thank you, Faraz. All right, moving on to number 8. I'm not going to read this passage because this is actually a question we've already answered before, but I am going to read this part down here. So base your answer to the question on the passage and on your knowledge of biology. A set of axes is shown in the diagram. When using this set of axes to show the effect of black walnut allelochemicals on the number of plants, which labels would be appropriate for axis X and axis Y? So this one required you to know which variable goes on which axis. All right, show me what you got in 3, 2, and 1. All right, can I hear from Baylin, please? Baylin, are you there?
Me?
Yeah, go for it.
Okay. So like we've said before, that the IV and the DV always have to be on, well, one of them always has to be on the Y and one of them always has to be on the X. And then I already knew that the two variables were the distance from the walnut tree trunk and the number of plants. And so then I was like, okay, the X is the IV and the Y is the DV. And so because the IV always has to be X, that means that the distance from the walnut tree has to be on the bottom of has to be X.
Absolutely. So using this sentence that I've highlighted, which is your IV? Is it the effect of black walnut, or I'm sorry, is it black walnut allelochemicals, or is it the number of plants? Which is your IV?
Black walnut things?
Yeah. So the black walnuts are going to be your IV, and what's the DV?
Number of plants.
Yeah, absolutely. So then using that information, we could plug that into what you said, where your IV is always on your x-axis, your DV is always on your y, giving us that the answer is 2. All right. Does anyone have any questions about that one? Because that can be tricky. Thanks, Baylin. All right, moving on to number 9. In an investigation, students determine the average rate of movement of gill covers of a species of freshwater fish at different temperatures. The results are shown in the data table below. Which labeled axes should be used to graph the relationship shown between the two variables? Show me what you got in 3, 2, and 1. All right, can I hear from Crystal for this one, please?
I chose 1 because if like it says the average rate of movement of the gills and the fresh water, so I knew that the gills was going to be on the y-axis and the temperature was going to be on the x-axis.
Perfect. Okay, so you said that gill covers are on your y and temperatures are on your x. So which is our independent variable here?
The independent variable is the one on the y-axis.
Flip it.
Oh, the x-axis?
X-axis, right? So temperatures are independent. What is our dependent variable?
The movement of gills.
Exactly. And so temperatures are IV, average rate of movement is our DV. So using that information that Crystal gave us, we're able to determine that the answer is 1, where we have temperature on the x-axis and the rate of movement on the y-axis. Great job. All right, moving on to number 10. This one has to do with the microscope. Now, this microscope does look a little bit different than the one we used, but you should have been able to use some context clues. Base your answer on the diagram below of a microscope and on your knowledge of biology. The highest possible magnification that can be obtained when using this microscope is 1, 40x, 2, 100x, 3, 400x, or 4, 4000x. Show me what you got in 3, 2, and 1. All right, Ariana, have I called on you yet today? I don't think I have. I have? All right, let me hear from Samaya, go for it.
Okay, so it says the highest possible magnification, and to me that means like the total magnification, the highest and the total magnification. So I looked at the the high objective power, which was 40x, and I automatically know that the eyepiece is 10x. So I multiplied 10x by 40x, and I got 400x.
Perfect. So your total magnification was 400x here. We're always going to use this formula. The total magnification equals the eyepiece times the objective. Okay, so in this case, it was 40x times 10x, which gave us 400x. Great job. All right, number 11. While viewing a specimen under high power, a student noticed that the specimen was out of focus. Which part of the microscope should the student use to obtain a clear image? Show me what you got in 3, 2, and 1. All right, can I hear from Arpita, please?
The answer is B, because B is defined focus, which will make the image more clear.
Perfect. So B here is our fine adjustment. What is this one called right here? Can someone tell me?
Coarse adjustment.
Coarse adjustment. All right, and we usually find these two pieces down here. But in this image, it looks a little bit different. But Arpita is correct. If we want to make the image more in focus and obtain a clearer image, we're going to use that fine adjustment, which was B. Great job. Number 12. A student sees the image below when observing the letter F with the low power objective lens of a microscope. Which diagram below most closely resembles the image the student will see after switching to high power? Show me what you got in 3, 2, and 1. All right, can I hear from Selena for this one, please?
I chose 1 because it was the same position as the letters shown in the image, just more clear.
Yeah, so when you switch to high power, it's going to make it more clear and more magnified. So the correct answer here was number 1, where we just see that F get bigger.
I have a question.
Go for it.
Isn't it supposed to be after you look, it's supposed to like rotate 180 degrees?
So that's a good
Yeah,
point.
that's why I messed up on this question, because when we were doing the lab yesterday, it flipped 180 degrees.
Yeah, I was debating on that.
It did flip.
Isn't that like it only flips 180? Like if that's only if like we're like seeing movement from the microscope, then it's like the opposite. It's 180, isn't it?
So you guys are all correct, but the key here was that we were already viewing it through a microscope. Okay, so this is
Yeah, that what got me 1. That's how I figured it out.
That's smart.
low power, and this is what the student saw in high power. Now, if it hadn't said that we were already viewing it through a microscope, then it would have been flipped, right? Because if this is our normal image, then our microscope image is going to look like this.
Mm.
Okay, but since we were already viewing it through a microscope, all we were doing was magnifying that image and zooming in. Does that answer your question?
Yes.
Yeah. Okay, so Crystal says, I don't get why it flips. It has to do with the projection of the lenses, right? It's a mirrored image because light is bouncing off of different magnifications or different lenses. So you're actually seeing a flipped image based on how that image is passed around. Now, this has to do with physics, and physics is not my strongest point, I'll be quite honest. But it has to do with the way that the image is flipped. Like if you have multiple mirrors, then it's going to be backwards because of the lens, exactly. That was, it took me a while to get there, but I'm glad that made sense. All right, any questions about that? All right, moving on to number 13. Just a few more questions, so power through. A new concept that is tested in a scientific investigation is known as a what? Number 1, theory, 2, hypothesis, 3, inference, or 4, observation. Show me what you got in 3, 2, and 1. All right, let me hear from Victoria, please.
I got 2 for this question because usually any concept that's being tested in an experiment is a hypothesis that they've created after doing research based on a question.
Beautiful answer, right? Your hypothesis is what you are testing in your experiment. So that is the answer. All right, let's power through number 14. Let's go a little bit fast because we're running out of time. Number 14, graphs of the data from laboratory investigations are used to 1, observe general trends in the data, 2, make the observed data more accurate, 3, prevent errors in measuring data, or 4, help change the original data tables. Let me hear from Miles. What'd you get?
For 15?
Or 14, I'm sorry. This one right here. Right, I can't hear what Miles is saying. Can someone else hop in?
One.
All right, and why is the answer 1?
Because you can't change the graphs of data, so you just have to observe it.
Absolutely, right? So using process of elimination, we use graphs to observe general trends. We can't make the data more accurate or prevent errors or change the data using those graphs. Your answer here was 1. Remember, I told you guys, whenever you're making a title, it's always going to be the effect of blank on blank. So in this case, our IV was temperature, DV was gill covering. It's the effect of the effect of temperature on the rate of respiration. All right, remember, test tomorrow. If you have any questions, please reach out to me and I will answer your questions. But other than that, have a wonderful day and I'll see you guys tomorrow. Good luck studying.
Bye, Ms. Collins. Enjoy your day.
Bye, have a good day.
Bye, Ms. Collins. Have a good day.
Bye, Miss Collins.
Bye.
Bye-bye.